Welcome to the Wright Science Daily Revision Boost. Today's Biology Blast is looking at decomposition and also the first part of genetics. So first thing I'm going to have a look at are decomposers versus detritivores. So these are two words we do need to know the difference between. When we're talking about a decomposer, these are microorganisms, bacteria or fungi, that break down or decay dead organic material as well as the urine and the feces, and as a result of this it returns minerals to the soil. So one phrase that we need to know is this word saprophyte. So saprophytes feed on dead material or waste by releasing enzymes onto the surface. Those enzymes digest the material and then the organism absorbs just the nutrients. When we're talking about a detritivore, these are small animals, and what they do is speed up decomposition by breaking organic material up into very small pieces. So as a result, the decomposers have a much larger surface area to work on. Three examples of detritivores to remember: earthworms, which will break down leaves, woodlice, which are going to break down the wood, and maggots, which tend to focus on animal material. If you're doing GCSE biology, we need to know the factors that will affect the rate of decomposition. So to increase the rate of decomposition, we need warm temperatures, a moist environment, and plenty of oxygen. And if they ask you to calculate the rate of decay, then it's the change in mass divided by the time that it's taken. So our second topic is about variation. So when we're talking about variation, these are differences within a species. And what we tend to see are the phenotypes, which are the observable characteristics of an organism. So we've got two categories of variation: genetic or environmental. So genetic variation caused by the genes you've inherited from your parents, and that's things like your blood group and eye colour, whereas environmental variation is caused by the environment in which you live, things like your accent or scars. However, don't forget we have some characteristics that are also caused by both the environmental and genetic variations. So this is things like your intelligence level and your height. When it comes to considering how we can actually display our data on variation, variations are either continuous or discontinuous. The continuous ones can take any value within a range, and these are caused by genetic and environmental variation. They'll be controlled by multiple genes, and some good examples here are skin colour, length of fur, height, etc. So because they can take any value within a range, we display that on a histogram, which, remember, are the ones where the bars all touch each other. If we're talking discontinuous, then this means it can take discrete values only. So this is where you would be either male or female. So it's caused by genetic variation and controlled by one or very few genes. Because it's discrete values we're getting here, we display it on a bar chart, which means there are gaps between the bars. Just go careful with the histogram versus bar chart, because it wouldn't surprise me if they were to give you a one marker or even potentially drawing a graph. If we consider the two types of variation we've got, then the first one, asexual reproduction, results in clones. And a clone is an organism that's genetically identical to its parent. So a good example of this is bacteria. They reproduce asexually, which means that all offspring are genetically identical to the parent cell. And this is done through a process called mitosis, which we've already looked at in our B1 to 3 paper. We've also got some plants that use asexual reproduction. Spider and strawberry plants send out little runners with baby plantlets on there. Potato plants use tubers, and daffodils use bulbs. For examples of animals that use asexual reproduction, we've got starfish and sea anemones. Second type of reproduction is sexual reproduction, which requires two parents. And this one will lead to genetic variation as DNA comes from both parents, so we're getting a mixing of genes. And this is the type of reproduction used by most animals. When we're thinking about sexual reproduction, if we talk about a gamete, that's a sex cell, so the sperm and the egg. Egg cells are made in the ovaries, sperm cells made in the testes, and gametes are made in a process called meiosis. So the gamete is haploid, which means they've got half the number of chromosomes of a normal body cell. Good examples to bear in mind are also plants here, because they also use sexual reproduction. So pollen is the male gamete, and that fuses with an egg cell, which is the female gamete, in the ovule to produce a seed. That seed then grows into our new plant. For those of you doing GCSE biology, you need to be able to compare asexual and sexual reproduction. So a handy little table here with those comparisons for you in terms of positives and negatives. Positives are green, negatives are pink. So the positives of our asexual reproduction is that if the parent is well adapted to the environment, the offspring will be too because they're clones, they're genetically identical, and only one parent is needed, so reproduction is faster. A downside to asexual though is that a change in a biotic or abiotic factor may destroy the entire species because they're all genetically identical. Positive of sexual variation, it leads to adaptations through variation, whereas a downside is that two parents are needed, which means reproduction is slower. And do remember we have some organisms that use both sexual and asexual reproduction to cover all bases, like the sea anemones. First topic for our chemistry check then is the reactivity of elements. Hopefully we remember that metals form positive ions, and the more easily this happens, the more reactive a metal is. So metals can react with water or acids if it's more reactive than hydrogen. So there's two general word equations in the middle there that we need to remember. Metal plus water makes the metal hydroxide and hydrogen, and metal plus acid makes the salt and hydrogen, because they could ask you to write the word or even balance symbol equations for those reactions, so you need to know what's formed. In reactions where a gas is made, the volume of the gas produced in a certain time can be used to actually work out the rate of the reaction. So the faster the rate, the more gas we make. And remember, a very accurate way of collecting this gas is with a gas syringe. But if they're giving you a practical to obviously analyse, then you could have an upturned measuring cylinder and then they ask you how you could improve it. So your improvement is use a gas syringe. One of the key reactions that we've already looked at in terms of the halogens are displacement reactions, but these also happen with metals. So in a displacement reaction, a more reactive metal displaces the less reactive metal from solutions of its compounds. So we've got our reactivity series on the right there with potassium at the very top of it and platinum at the very bottom. Remember, hydrogen's not a metal, but it's in there to give you that reference point for our metal and water and metal and acid reactions. So in terms of our half equations, then if we've got the reaction at the top there, which is copper reacting with silver nitrate, then the half equation for copper is going to be our solid copper makes copper ions and 2 electrons. Remember, electrons have the E- symbol, which is an oxidation reaction. And our silver ions gain 1 electron to become silver atoms, which is a reduction reaction. Don't forget, good old oil rig, oxidation is loss of electrons, reduction is gain of electrons. And because we've got both oxidation and reduction, it's a redox reaction. A few useful patterns that are worth remembering here. If we're talking group 0, they don't react because they're inert. Group 1 and 2 become more reactive as you go down the group, whereas group 7 becomes less reactive as you go down the group. Group 1 and 2 are more reactive than the transition metals and other metals. If we're talking about metals, they may form ionic compounds with reactive nonmetals, and reactive nonmetals may form covalent compounds with each other. So it's going back to our ionic bonding between a metal and a nonmetal, and then covalent bonding between nonmetals. If you're doing GCSE chemistry, then we need to know instrumental methods of analysis, which is our second part of today's chemistry check. So instrumental methods of analysis rely on machines to carry out the analysis on a substance. 3 key advantages we should remember. It's got a high degree of accuracy, it's very fast and can run 24 hours a day, 7 days a week, and it's very sensitive, so it can analyse very small amounts of samples. Hopefully we remember the process of gas chromatography from our earlier chemistry topic in C2. So remember, we inject the sample, it turns into a gas, and the carried gas pushes it through the coil and then is detected by the detector as it comes out, creating our chromatogram on the right. And remember, the gas chromatogram is a chart that represents the different substances in a mixture, and the height of the peak tells us the strength of the response. So if you're asked to analyse a chromatogram, then each peak is a substance, and the area under the peak shows the relative amount of the substance. So what we've got on the bottom is their retention time, and that's different for each substance, hence the different peaks for each one. So the retention time is just the time it takes for the substance to travel through the chromatography column. The last process we're looking at is mass spectrometry. So mass spectrometers measure the masses of atoms and molecules. So we use them to analyse the relative amounts of isotopes and the structure of different molecules. So when a sample enters the mass spec, then it's ionised to form molecular ions. As these molecular ions are gaining energy, then they may fragment, so break up into smaller pieces. And as these ions then pass through the machine, the detector records the amount of each fragment that we've got. Picture on the right is our mass spectrum then. So the peak on the far right is the molecular ion peak. So that is our relative formula mass for the actual molecule. So if they ask you to identify it, look for the peak on the far right, and that gives you your relative formula mass for the whole molecule. So what we then have, on the ones on the left there, those are the fragments of the molecules, and the height of the peak tells us the relative amounts of them once more. Today's physics prep is going to be looking at electromagnetic waves. So hopefully we know that when white light passes through a prism, we can see the visible light spectrum because it splits it. So visible light is just one part of our electromagnetic spectrum which we need to remember. So radio, micro, infrared, visible, ultraviolet, X-rays and gamma rays. Remember them in sequence and remember which ones have the longest wavelength and the highest frequencies, because these are going to help you out in answering questions. We should also remember that all electromagnetic waves travel through a vacuum at 3.0 times 10 to the power 8 meters per second. And that's also the speed of light because it's part of the electromagnetic spectrum. Do remember that when we're referring to a source, then these are ones that emit electromagnetic waves, but we've also got other objects which will absorb electromagnetic waves, like your skin, for example. If you're doing the higher tier, you need to know about how radio waves are produced and detected. So what we end up with is an oscillating potential difference across a wire, which makes the electrons move backwards and forwards. As a result, we've got a changing electric and magnetic field being produced, and that's what's emitted as our radio wave. When those fields then meet a piece of metal, which is your big antenna, then the electrons move and an electrical signal is produced. We need to know some uses of electromagnetic radiation. So microwaves are used in mobile phones, satellites, Wi-Fi and Bluetooth. Radio waves are used in radio and TV. And infrared is used in your remote controls and optical fibres. And we can also use visible light to communicate between ships. So our first little block there is communication links. We've also got the ability to use lasers, which are using visible light to read discs in CDs, DVDs, Blu-rays, etc. And then in terms of some of these other uses, we've got our body using ultraviolet radiation to produce the vitamin D to avoid rickets. We've got forensics officers use UV light to identify forged banknotes or body fluids. And UV light can also be used to sterilise water by killing the bacteria. In terms of medical uses, X-rays can kill cancer cells and also produce images of bones, whereas gamma rays can kill bacteria and kill cancer cells. One thing they used to like was a question about the difference between cooking using microwaves and infrared radiation. So we know electromagnetic waves can be used to transfer energy, and microwaves are one example of these. So your microwave oven emits microwaves which are absorbed by the fat and water in the food, and they're actually able to penetrate a few centimetres in. From those first few centimetres in all the way around, energy is then transferred to conduction by the middle. If we compare that to a conventional oven, then they use infrared radiation. And infrared radiation is only absorbed by the particles on the very surface of the food. And then conduction has to transfer that energy through the rest of the food to cook it. So because microwaves penetrate further into the food, they're faster at cooking than a conventional oven. Last thing to look at are the dangers of EM radiation. So ultraviolet light can actually damage the DNA in skin cells, leading to skin cancer, and too much exposure to UV in your eyes can actually cause cataracts. In terms of our X-rays, they can damage cells and cause cancer. A bit contradictory there as we use them to treat cancer too, but there we go. And so radiographers will stand behind a LED screen or leave the room to reduce their exposure. So they also wear a little badge that tells them when their exposure limit has been reached. Last ones are gamma rays. They can also kill or damage cells in the body too. For those of you doing GCSE physics, you do need to know how we use EM waves to image. So thermal imaging cameras produce a thermogram, and basically we've got pixels inside a CCD, a charge coupled device, that absorb infrared radiation and produce the image. Those then have colours added by a computer so that hotter areas emit more infrared radiation, which we colour in the reds and the oranges. If we're talking X-rays, then bones absorb X-rays but soft tissue does not, so that if we place photographic film down, it absorbs X-rays as it darkens. And so what we actually see there is the bones are showed as a whiter colour because we've not had the X-rays absorbed in that part of the film. We can also use a CCD to detect X-rays, and the colours show the different densities of the material. So the higher the density, the more X-rays are absorbed, and we can produce these CT or computerized tomography scans that allow us to look inside the body without cutting it up. In terms of our gamma rays, we can use these as tracers in medicine. So the tracer is injected, swallowed or inhaled, and then we use a scanner to detect the gamma rays that are emitted from inside the body. And then we can see those on a computer screen to identify blockages or leaks, etc. Do remember when we're talking about gamma rays, we can also use them as tracers in underground pipes. So if we get a greater concentration of gamma rays at one point, that's the source of the leak. Now we've had a quick recap, then head on over to socrative.com and test your knowledge on the topics from today. The room name you need to enter is Wright 2662. Good luck! If there's been any areas in today's little booster that you've not been too sure on, head on over to the playlist to get a more detailed look at those topics. So for biology, it was B4 and the start of B5. Chemistry was C4. And for the physics, if you're doing combined science, it's P4, whereas if you're doing the separate sciences, it's P5.